Hello everyone, we are from Group 4. Today we'll be presenting on APOMIXIS. These are my group members. So what is APOMIXIS? It stands from two Greek words, apo which means away from and misis which means act of mixing. When combined together, it means the act of not mixing. APOMIXIS is a naturally occurring mode of asexual reproduction in flowering plants that results in the seed formation without the involvement of meiosis or egg fertilization. The seedlings produced through this manner are called apomixed, and they are identical to their mother plant as they have the same genetic makeup as that of the mother plant. This is clonal seed production. The pictures on the left are some of the examples of apomictic plants, which are citrus, dandelion, and hawthorn. Winkler defined apomixis as the replacement of the normal sexual reproduction by asexual reproduction without fertilization. This is because apomixis mimics the sexual reproduction of plants. The discovery of the first apomictic plant was by Leeuwenhoek in citrus seeds back in 1791. In apomictic species, the sexual reproduction may be suppressed or absent. In cases where sexual reproduction occurs, it is known as facultative apomixis, and when sexual reproduction is absent, it is called obligate apomixis. One of the examples of facultative apomixis is bahia grass, and for obligate apomixis is sea buckthorn. So this is how we determine apomixis. In this example, homozygous dominant plants are red and homozygous recessive are white in color. A group of homozygous dominant and homozygous recessive plants are put together whereby the female plants are homozygous recessive. If the F1 generation produces progeny that are white traits, then it indicates apomixis, otherwise they undergo normal sexual reproduction. Since fertilization does not occur, the progeny will follow the mother's trait. Apomixis is sometimes confused with parthenocapy. There are both asexual reproduction that do not undergo fertilization, producing offspring similar to the parents. Parthenocapy develops seedless fruits and is seen in plants and animals. In parthenocapy, the plants are usually induced by growth regulators such as auxin and gibberellin. Apomixis, on the other hand, develops seeds without fertilization and is seen in angiosperms and gymnosperms. Apomixis also mimics sexual reproduction and occurs naturally.
Next, I'm going to explain about the types of apomixis. As we all know, in usual sexual reproduction in plants, the seed germinates after the fertilization occurs after the fertilization of a male gamete and female gamete. So the male gamete and female gamete will fuse and form zygote, diploid zygote, and from the diploid zygote, the zygote will develop into embryo, and the ovule becomes seed, and the ovary becomes the fruit. But in apomixis, the seed germinates without fertilization, and in some cases, the seed germinates without fertilization and also without meiosis. So let's see what are the recurrent and non-recurrent type of apomixis. First is recurrent. Recurrent is temporary substitution of sexual reproduction, where in some cases, in some plants, the sexual reproduction is not, the conditions are not favorable for sexual reproduction. In this situation, the certain plant will change its reproduction system from usual sexual reproduction amphimixis to the temporary substitution which is the apomixis. But for the non-recurrent apomixis, this is a permanent way of reproduction system in certain plants. In this case, the meiosis will take place, but the fertilization will not occur for the production of seed. So as I mentioned, there are two types of recurrent apomixis. The first one is vegetative reproduction. We all know what is vegetative reproduction. So this vegetative reproduction will substitute the sexual reproduction in unfavorable condition. So the next recurrent apomixis is agamospermy. So this agamospermy is a type of asexual reproduction in which the plant propagation occurs by apomictic seed. So this apomictic seed is the seed that produced without fertilization. So there are two ways of production of seed in agamospermy. The first one is diplospory. Diploid embryo is formed from megaspore mother cell by mitotic division or by interrupting meiosis. In usual production of haploid egg cell, the megaspore mother cell will divide meiotically and will produce 8 haploid cells in the embryo sac. One of the 8 haploid cell is the egg cell. But in this case, in diplospory, the megaspore mother cell will not undergo meiosis, but it will undergo mitotic division and will form unreduced embryo sac. So the seed will produce from this unreduced embryo sac, which is in diploid state. In apospory, the nucellar cell divides into diploid embryo sac. So what are the nucellar cells? Nucellar cells are the cells that fills the space inside the ovules. The megaspore is also placed within these nucellar cells. So in this apospory, the diploid nucellar cells will will undergo mitotic division and will produce diploid apomictic embryo sac, not the megaspore mother cell. Now we see what is the non-recurrent apomixis. In non-recurrent apomixis, the megaspore mother cell will undergo usual meiotic division and forms haploid embryonic cell. But in this non-recurrent apomixis, the fertilization will never occur. So in non-recurrent apomixis, there are two ways of seed production. One is parthenogenesis and another one is apogamy. So what are the difference between these two types of non-recurrent apomixis? As I said before, in this recurrent apomixis, there is meiosis, where this megaspore mother cell will undergo usual meiotic division and will form 8 haploid embryonic cell. But what are the difference between parthenogenesis and apogamy? In parthenogenesis, when the embryo develops directly from haploid egg cell and produce it, that is called parthenogenesis. But for the apogamy, when the embryo develops directly from some other haploid cell other than the egg cell of the embryo sac, is called apogamy. Last type of apomixis is adventive embryony. Embryos are produced directly from nucellus or integument of the ovules. We already know what is nucellus, but the integument is the tissue that protects the ovules. It covers the ovule. In this adventive embryony, the embryos develops by mitotic division and forms bud-like structure. So the cells of integument or nucellus are diploid. So obviously the embryo that produce are also diploid.
Next, I will be talking about the evolution of apomixis. Research over the last century has provided some insights into the evolution of apomixis. It has shown that agamospermy, like other features of the breeding system of plants, is under genetic control and is usually governed by 2 or more loci. However, the whole question of evolution of sexual versus asexual reproduction remains a highly controversial area of research. This controversy is reflected in the fact that three contrasting viewpoints have appeared in the literature regarding the adaptive significance of apomixis and its establishment in plant populations. They are the escape from sterility hypothesis, Henry Ford or Model T hypothesis, and the automatic advantage hypothesis. The escape from sterility hypothesis was first proposed by Darlington in 1939 and promoted by Stebbins from 1941 to 1950. Agamospermy offers no real selective advantage over normal sexual reproduction. It restricts recombination and hence the evolutionary potential in species in which it becomes established. From this viewpoint, the principle states that the adaptive advantage of apomixis restores the fertility of individuals that would otherwise be sexually sterile because of polyploidy and hybridization. This theory achieved wide currency because it was compatible with the prevalent view that sexual reproduction greatly accelerated the rate of adaptive evolution and was maintained in species by group selection. The second one is the Henry Ford or Model T hypothesis. It was proposed by Clausen in 1954. He was among the first to recognize formally that apomixis, particularly facultative apomixis, which does not necessarily lead to a loss of variation and evolutionary potential. He argued that the combination of sexuality, which allows for the progressive production of new genotypes, and apomixis that allows for the unlimited and faithful reproduction of the best of the genotypes generated. The hypothesis that partial apomixis offered more than a short-term escape from sterility was also widely accepted. It is supported by findings that the level of apomixis in several facultative species varies, presumably adapted with environmental conditions. Last but not least is the automatic advantage hypothesis. This hypothesis developed from recent quantitative studies designed to resolve the highly controversial question of the evolution of sex. These studies shows that asexual reproduction has an automatic twofold advantage over sexual reproduction, which can accrue because of either within individuals or group selection. The twofold advantage of asexual reproduction at the individual level arises because of all the offspring of a parthenogenetic or apomictic parent that carries the full genetic complement of their mother. Sexual outgrowth offspring will carry only half the genetic complement of their maternal parent.
In this segment, the overall mechanism or perhaps the establishment of apomixis in plants are summarized to three phases. It is a fact that apomixis is in general rare and still requires further research and studies to confirm those multiple hypotheses regarding the formation of apomitic individuals, populations, and finally the niche or geographic distribution of its genetics. Foundational phase explains those theories regarding the formation of an apomictic individual. In this aspect, the foremost seed formation origin are regarded in two ways, that is either dispersed from an already apomitic source or a spontaneous shift to apomixis in a normal sexual reproducing population. Traditionally, hybridization is the key hypothesis that acts as a trigger for the emergence of apomixis. Hybridization affects only the formation of an unreduced embryo sac and do not involve in parthenogenesis and endosperm formation of apomictic seeds. Another well-reserved process is polyploidy, which results in genomic shock that leads to expression of apomixis, but rather artificially. In terms of environmental condition, that also includes pollination timing or a low stress condition in polyploid tissues that stimulates the sexual meiotic pathway, thereby releasing the inherent potential of plants for apomeiosis. Besides that, endosperm formation is under a different epigenetic control and is dependent of fertilization of polar nuclei, pseudogamy in most natural apomix. Therefore, the endosperm and parthenogenesis loci are linked but separate. This overall need for multiple factors in those three development stages makes a mutagenic origin of apomixis naturally very unlikely. After that, the establishment phase or simply the formation of apomictic population, whereby the uncoupled expression of apomixis leads to the formation of polyploid population as a survivability of the lineage. Thus, uncoupled activation of apomeiosis and parthenogenesis in a diploid cytotype will increase the ploidy which stabilizes the expression of apomixis elements. In natural conditions, this mostly happens through a triploid intermediary that facilitates the formation of even polyploids, like in sexual systems. In some apomictic systems, sexual cytotypes are polyploid and no evidence of occurrence of diploid cytotypes is found in nature. This may be due to sexual diploids that first undergoes the sexual polyploidization via unreduced male gametes that fertilizes the reduced egg cells and then become extinct. Polyploidization comes with a side effect such as the breakdown of SC systems, resulting in self-fertility. This self-fertility with pseudogamy further avoids the negative effects of sexual selfing, namely loss of heterozygosity and inbreeding depression. Self-fertility is further beneficial for founding new population by single or few founders even after long distance or dispersal of seed. Under natural condition, functional apomictic seed formation in diploids probably requires certain altered ecological condition and successful polyploidization for establishment. Finally, once an apomictic population is successfully established, its survival will sure depend on the neopolyploid's capacity to either outcompete the parental diploids or migrate to another habitat. These non-hybrid apomix do not show a pronounced genetic diversification, but nevertheless manage to occupy habitats outside the ecological range of the diploids. Self-fertility and apomixis enables the plants to form a new population starting with a single individual. On the one hand, apomixis allows for founder events and spread of species populations after seed dispersal. On the other hand, apomix creates the clonal offspring and hence it multiplies. This double advantages provides plants with a better colonizing abilities and both have a relevant impact on genetic variation and population level. Morphological differentiation is observed within apomictic complexes, whereby facultative apomixis in hybrid species can promote divergent selection and formation of microspecies. Alternatively, a shift back to a sexual reproduction in a distant population from the parental sexual species of the polyploid apomix would also allow for independent evolution and morphological changes. This summarizes the entire tree phase of an establishment of apomixis in a plant system.
Next, I am going to talk about the genetics and inheritance of apomixis. Before I begin, I want to highlight that the genes responsible for apomixis are not completely genetically characterized yet, due to the complexity of phenotypic screens and the differences of apomix reproductive systems. Generally, apomixis is inherited as a simple dominant trait controlled by a few independent loci. It is once thought that apomixis was inherited in a single locus. Now, it is known that it involves a number of loci. For instance, all three loci that are not linked have to be inherited by the progeny to express the complete autonomous oposporous of apomixis in Hericium species and autonomous diplosporous of apomixis in Hericium species. It is proven that meiotic avoidance, parthenogenesis, and fertilization-independent endosperm development are regulated by independent loci. For example, Hericium and Erigan species showed control of diplospory and parthenogenesis by independent loci. It is observed that the loci controlling apomixis has low rates of recombination, leading to hemizygosity in extreme cases where the alleles have diverged significantly. This leads to the speculations of apomitic genes are present in supernumerary chromosomes. Suppressed recombination is associated with a heterochromatic structure of the chromosomes, a lot of repetitive sequences and transposons, which causes large-scale chromosomal rearrangements. There are two explanations for these repeated sequences. First, they are hypothesized to act as a sink to hide away factors controlling sexual reproduction, thus allowing apomixis to occur. Second, if their presence is not involved in functions leading to apomixis, the unique chromosomal and genetic structural features and allele divergence are caused by asexual reproduction and suppressed recombinations, which have evolved and are important to maintain the elements needed for apomixis. Studying differentially expressed genes at apomix and sexually reproductive plants could provide insights on what apomixis-related genes and pathways are involved. Transcriptome comparison analysis between apomitic and closely related sexual individuals from reproductive tissues at different developmental stages have been carried out. It uncovered pathways related to apomixis. There are protein degradation, transcription, stress response, and cell-to-cell signaling. For example, it is shown that expression for stress-induced proteins at oposporous initial cells suggested that apomixis might be induced by stress. Epigenetics are starting to gain researchers' interest in understanding apomixis. Few experiments have indicated the presence of epigenetic regulation of apomixis, although it still requires much more confirmation. However, this shows that polyploidization of individual plants might be followed by epigenetic changes, consequently leading to activation of apomixis and suppression of sexual reproduction. Thus, this explains the reversibility of apomixis to sexual reproduction in some plants and facultative apomix.
Next, we are going to talk about apomixis in agriculture and plant breeding. So apomixis complicates plant breeding. This is because in obligate apomix, which are not sexually reproducing, it is very hard to make crosses between them. However, we can do it, just that we need these three steps. First, restore sexual reproduction and stop apomixis. Now that they are sexually active, we make the desired crosses to gain the traits and the goals that we want. After that, we can re-establish apomixis. In non-apomix, which is naturally sexually reproducing plants, we can make them into apomix, and there are two methods. First is through transfer, where we interbreed them with wild species that have apomixis, so that these sexual plants, they gain apomixis. And another is synthesis, where we mutate them to the point that they have apomixis. So what are the benefits of apomixis in agriculture? It is cheap cloning. So we are able to produce progeny with 100% the same genetics without requiring expensive equipment or a lot of time. And the seeds produced are easier to store than plantlets like we normally see in cloning. We are able to maintain crops with desirable traits. This is because we are able to fix heterosis and stabilize segregation in hybrids, which in turn provides crops with high yield. Now, apomixis introduces a higher efficiency because the females do not require males to be present, which is the pollinators. So the seeds are able to be instantly produced once it reaches maturity, and it doesn't need male reproductive organs, which further increases efficiency. Now here are some examples of apomictic plants. We have here the group citrus, which are apomictic mainly through nucella embryony. Examples are the lemon, lime, sweet and sour orange, mandarin, and grapefruit. However, the citron and pomelo are sexual. Next, we have the fruit trees and berry bushes. We have the mango, mangosteen, and walnut, which reproduce through adventative embryony. We also have blackberries and strawberries. And we also have apomictic crab apples, which is a group that includes the apples that we normally eat. And they reproduce through facultative apomixis. And one of the most important economic plants is the rubber plant, which includes Chondrilla, Taraxacum, and Parthenium. And here are our references. And that's all from us. Thank you so much.